before heading to our debate session, I want to inform you about what is the CRISPR technology. And CRISPR is basically gene editing and discovered in 2012 in USA. CRISPR, a lot of scientists mention about the CRISPR, the most important invention in whole humanity. And basically, we can change our genes and we can end of our sickness. We can get rid of the old sickness, and some futurists say with this technology, we can find immortality as soon. So it is really important. And in 2020, in this year, Emmanuel Charpentier and Jennifer Doudna won Nobel Prize with this project. So let's heading to our debate session, and we will start with Malaysia. Hello, Malaysia. How are you?
Hello.
Hello, Miss Muchami.
Yeah.
Are you gonna be the one who present?
Yes, for CRISPR, I'll present.
Okay, I'm giving you hosting, and when this ends, could you give me back, please?
Yeah, sure.
Thank you very much. Thank you very much. Thank you.
So good day, everyone. I will share my screen, and I hope you could lend your ears and eyes also mind to enjoy this session.
Okay, good day, everyone. Usha and myself will be presenting about the ethical dimension for the commercial research and the development of CRISPR Cas9 technology. Okay, so let me introduce the CRISPR technology and the mechanism of it. Cluster regularly interspace short palindromic repeat, which is known as CRISPR, is the family of DNA sequence that can be found in the genome of prokaryotes, like the bacteria and the archaea. Okay, the CRISPR-Cas9 technology is the latest genome editing tool, which is simple, yet powerful and precise. It involves the altering and the modification of the targeted gene sequence with the aid of a protein, which is known as Cas9. Okay. The Cas9 is a molecular scissor that cuts the DNA strands. This concept can be found in the virus-attacked prokaryotes. These prokaryotes store in a specific DNA sequence of the viruses and use it as a tool to defend itself from the second attack by editing its own genome. Okay. Okay, each and every DNA strands will contain the targeted sequence that we are looking for. First of all, the guide RNA binds to the DNA sequence, and it will have the Cas9 enzyme to bind to it, and the Cas9 enzyme will cut both of the strands of the DNA. Okay, let me show you a video that explaining the mechanism of the Cas9 technology.
So actually, there was supposed to be some sound, but due to technical errors, the sound wasn't be heard. So I will move forward, which is the next part, which is the commercial research and development. While CRISPR-Cas9 can be used in various industries or fields, and one of most popular industries is healthcare. So CRISPR-Cas9 can be used to treat cancer, to treat genetic diseases such as sickle cell anemia, and also organ transplantation. Well, actually, sickle cell anemia is the red blood cells, which is shaped in a wrong way. So the next is food industries. Well, for food industries, we could produce dairy products such as yogurt and cheese through CRISPR-Cas9 technology. And it gives a resistance for these yogurts and cheese towards the infection. And the third one is agriculture. CRISPR-Cas9 can be used to increase the yield of crops and to control the pest invention. Well, I would like to add something, how CRISPR-Cas9 control the pest invention. There is a bacterium called Bacillus thuringiensis, and it produces a protein that repels insects. So these are the industries that uses or applies CRISPR-Cas9 technology. Well, from the ethical dimension of CRISPR-Cas9 technology, the first thing is honesty and integrity. We have to ensure that all the data related to the studies or research being done must be manipulated honestly. So there shouldn't be any addition or removal of data on the study. The next part is carefulness. We should be very aware of the environment and be meticulous. Well, we have to ensure that every study or experiment being carried out carefully. And the third one is legality. Using this CRISPR-Cas9 technology, we should be aware towards the rules and regulation enforced by the government. So we always have to obey the rules and regulation. And the last one would be nature law. We are aware that we have a law of nature. So every study should be conducted by taking the nature and its interaction into account. So anything that disobeys the natural law should be given more attention. And one of the crucial risks of this CRISPR-Cas9 technology is the off-target effect. Well, off-target effect is that the Cas9 protein cuts the wrong DNA sequence, because we can't assure that the Cas9 protein will cut the correct base sequence in the DNA. It might mismatch. So this can cause mutation. And the worst thing is when we use this technology in germline cells, which is the reproductive cells, it can cause a lot of mutations for the future generation. Although there are some good points in that, but we should always ensure that we are not affecting the future generation. And there was a study done by Chinese researchers led by Junji Huang, and out of 6 embryos being experimented, only one survived the beta thalassemia-free DNA cells. So, and 2 of it was the DNA sequence was corrected. However, it had the mutation, which causes the off-target effect. And we should be very aware when using CRISPR-Cas9 technology towards germline cells and enhancing the embryonic studies. So these are our references. And thank you, everyone. And I hope that you got the idea of the ethical dimension that involves in this CRISPR-Cas9 technology. And also this technology can be used to fight COVID-19. So let's support our researchers to find or to make a vaccine as soon as possible. Thank you.
That was great presentation, Ms. Ushami, and of course your friends and your teachers too. Thank you very much for the hosting again, by the way. So we are really, thank you very much. It was really enlightened us. Now we are heading to second presentation, which has come from our school, our country, Turkey. Now we are heading on our Turkish student, Başak and Derya, which is also my students too. Of course, we will, before we will get the presentation, thank you very much, Malaysia, again. And I want to say hello in my own language. Merhaba.
Merhaba.
Okay, let's hear from you, guys. Thank you very much.
Okay, so hello everyone. Firstly, I would like to open my presentation. Okay. So, hello everyone. This is Başak, and this is my friend Derya. Hi. So, to us, science is like a coin which has two sides. One side is the perfect, legendary, positive side, but the other side includes what can people do with that discovery for themselves. So right here today, we would like to show you these two sides of CRISPR. As you can see, we will walk through three different ways. The first one is the past. Actually, we will talk about what is CRISPR-Cas9 briefly and how does it work. In the second presentation, my friend
about stepping stones and what is the dream behind the CRISPR technology, and in the third we're talking about the future possibilities which we described as the infinity and beyond. Now let us begin.
Yes. So firstly, what is CRISPR-Cas9? As you know, genome is kind of like an instruction manual that makes us who we are. And sometimes there is mistakes in the instruction manual. So CRISPR is a tool that scientists are using to edit or change genomes. It is faster, cheaper, and more accurate than previous techniques of editing DNA and has a wide range of potential applications. It's a unique technology that enables medical researchers to edit parts of the genome by removing, adding, or altering sections of the DNA sequence. And as you can see from also the other presentation, put that same picture. As you can see, there's a target sequence and the RNA is coming. So we're not going to talk about that because
it's explained already. Now we want to start with when it started to become real. Firstly, alchemists. Now we have talked about what CRISPR is and how it works. And now we want to show the path behind CRISPR, CRISPR's ultimate dream, which we believe is eternal youth and immortality and cure of all diseases we know of. And all of these goals have been experimented firstly by alchemists. Now, who then set the ground for modern chemistry. What then seemed to be just a dream back then may be true within 100+ years with CRISPR's technology. Now, in second picture, we see Aldous Huxley. Now, there are many examples of novels that target the genetically modified future societies, but we saw fit to put Aldous Huxley's Brave New World because of its intelligence-based society and how it is separated into groups. And we believe that it is a similar dystopian idea that many of the scientists think is possible. And thirdly, of course, Mendel's researches. We are moving forward a couple hundred years from alchemists, and we see that Mendel's research in IVF treatments. The reaction he got was similar to the discussions of ethics in evolved CRISPR. Now, I want to further explain that one because one of the really nice things about CRISPR is that we can use it in human cells. If you ask most people, should you use it to cure cancer? Most people say yeah. Most people would say yeah. But there are other, those are good uses, of course, but there are other areas that are still stands a bit more problematic. Now, like editing the germline means something that could be passed off. Ethics-wise, a lot of people have that natural reaction of fear against something they don't know, something that is so new and has a potential, we just don't know where it can take us yet. And it was sort of similar to when people and Mendel coming first started to research and experiment on in vitro fertilization test tube babies. And that was kind of a similar scary situation for the public too. Now, moving forward.
Now you will watch a video about the benefits
Research into this
of
is
the
in its
CRISPR.
infancy. And many scientists are rightly skeptical about the end of aging. The challenges are enormous and maybe it is unachievable. But it is conceivable that people alive today might be the first to profit from effective anti-aging therapy. All we might need is for someone to convince a smart billionaire to make it their next problem to solve. On a bigger scale, we certainly could solve many problems by having a modified population. Engineered humans might be better equipped to cope with high-energy food, eliminating many diseases of civilization like obesity. In possession of a modified immune system, with a library of potential threats, we might become immune to most diseases that haunt us today. Even further into the future, we could engineer humans to be equipped for extended space travel and to cope with different conditions on other planets, which would be extremely helpful in keeping us alive in our hostile universe.
Okay, so the benefits, we will briefly mention the benefits and talk more about the possibilities in the future. So as you can see, malaria from mosquitoes and Alzheimer's diseases, HIV, new drugs, livestock, and cancer treatments are the few benefits of the CRISPR-Cas9 technology. I think an ethical component is if CRISPR does make it to market, who will have access to it? That is going to be cost prohibitive. Will only a certain select few be able to use it, as many new cancer drugs are today? Will people start using it for non-therapeutic reasons? And we think this, then can get into a whole slippery slope. A lot of people are concerned maybe CRISPR will be used in frivolous ways just to choose how tall they are, what color hair they have, and I think what a lot of people don't realize is the state of genetics is not quite there yet.
Yeah, it's nice to think that I can go in and change exactly what color I want my eyes to be or my hair to be. But we don't. But fundamentally, science doesn't exactly know all of that yet. We don't know where every single gene related to our eye color, nor the regulatory mechanisms, nor the epigenetics of it. So we can now use CRISPR to try and understand better how do our genes relate to these different features, phenotypes that we have. And there's a lot of work to do before we even know what are the knobs and dials and the controls that you know affect the eye color and how tall you are, etc. So the real question is that we need to ask now, even though it is important to discuss these topics, they are more rhetorical right now, or is it ever really going to be a point in will they be necessary for conversation? Because people are going out every single day, and getting plastic surgery to change how they look, and there is nothing wrong with that. And how is this any different than people using virtual fertilization, right? So this is manipulating genomes. You're choosing which embryos to implant, so we're almost in this little aspects of CRISPR has the potential to do. Now, there's another video, I believe, about dystopian features.
The means to edit the genome of a human embryo already exist, though the technology is still in its early stages. But it has already been attempted twice. In 2015 and 2016, Chinese scientists experimented with human embryos and were partially successful on their second attempt. They showed the enormous challenges we still face in gene editing embryos, but also that scientists are working on solving them. This is like the computer in the 70s. There will be better computers. Regardless of your personal take on genetic engineering, it will affect you. Modified humans could alter the genome of our entire species because their engineered traits will be passed on to their children and could spread over generations, slowly modifying the whole gene pool of humanity. It will start slowly. The first designer babies will not be overly designed. It's most likely that they will be created to eliminate a deadly genetic disease running in a family. As the technology progresses and gets more refined, more and more people may argue that not using genetic modification is unethical because it condemns children to preventable suffering and death and denies them the cure. But as soon as the first engineered kid is born, a door is opened that can't be closed anymore. Early on, vanity traits will mostly be left alone. But as genetic modification becomes more accepted and our knowledge of our genetic code enhances, the temptation will grow.
So we have talked about dystopian features now, and of course, one of the questions that's very interesting is trying to understand the effect of human genetic variation on using CRISPR-Cas9. Because you and I just sitting here, millions of differences within our genome. We use these guide RNAs and match it to different places in the genome. But what if there is a mutation at that side, and how does that affect a mutation that's different between you and I? It is the same side, but there's a slight difference in the DNA between us. Let's say in you, it's a perfect match. In me, there is one that's actually a mismatch. And how does that affect things in the lab? Because maybe it won't be as efficient. It won't be able to bring Cas9 to the correct side and cut that. There's another reason why there must be a lot more research to be made. And as you can see in the photos, there are designer babies and supernatural army, and the society will actually, we were going to talk about that. The society might discriminate people who cannot or their bodies are unable to accept CRISPR-Cas9 technology, and how they can or could be shunned from society in the not foreseeable, but from 100 or 50 years from now, etc.
And in the second picture, we talk about the science experiment. We're going to be, if someone who is going to use that for their benefits, they can make an army with this CRISPR-Cas9 technology, and they can use it for to kill humans.
Think it like a more advanced biological warfare idea, but beyond our imagination right now.
And there is another video that we want to show you. There is a doctor that's going to talk about the CRISPR-Cas9.
He's a professor in New York University, and he's talking with another professor, researcher from Harvard University. Oh, oopsies.
Okay.
Know very well from sequencing many different individuals, there are a lot of differences. And so there are some concerns that when you use these in the lab that you're intending for a cut to be made in this one place in the genome. But if there happens to be a mismatch there because of some variant in the cells that you're studying, maybe it won't cut there at all. And even maybe worse is that maybe it'll cut somewhere else because maybe now it can match elsewhere.
So you're saying right now that it's kind of early days with CRISPR, and that there's a lot of technical issues that really need to be ironed out, that it's not anything can be targeted at any time. We're trying to develop the scientific steps that will take us to that kind of total genome control.
It is still the early days, and as much progress has been done, which is actually quite remarkable given how new this technology is. I mean, we're just a few years away from when it was really started being used for these applications. However, having said that, it holds a lot of potential and I think can get there, but there's still a lot of issues and concerns that need to be worked out.
I was going to say that a lot of what inspired me to get into bioengineering are movies like Jurassic Park, which both shows the good and the bad sides of manipulating the nature around us. And so maybe it's a bit of a silly question, but what do you think in our lifetime will we see something like Jurassic Park where humans have actually engineered different animals?
Anything is possible. I'm not sure that we will see that in our lifetimes, but I would be very excited to attend Jurassic Park if that were the case. Having said that, I do think that CRISPR Cas9 does have a lot of potential to do things that are kind of out of the box.
Now, before that, more than anything, CRISPR has created a whole open field of opportunities. And so whether or not that leads us to Jurassic Park, there isn't a certain answer. Opportunities might be much broader than just biomedical science. It might be things like using DNA as a hard drive, being able to use CRISPR as a diagnostic instead of just a tool for editing, and we use cells to record data over time. It stands a possibility amongst many others. Now, can we use CRISPR to track cells in developing embryos? This is one of the most exciting recent uses of the dominant paradigm right now. CRISPR is just for cutting DNA. But when you think of it more as a general platform, a pointer to a location in a genome, then that becomes a very powerful thing.
So certainly a great goal is ever being at a place where based on looking someone's genome and these kinds of CRISPR functional screens, we can predict what kinds of diseases that people might get 10, 20 years in the future. I think that's as of we tend to focus on variations or mutations that we know the function of. But about mutations, we have never seen before something that comes from, let's say, you have radiation or another thing. We could ultimately have the power to do that. One of the ultimate goals, being able to correct the mutations for diseases we have known about for sometimes hundreds of years, and we just haven't been able to do something about it. But correcting these very old mutations can cause us to face mutations we have not seen before. As the end.
Now we have talked about what CRISPR is and how it works and the history behind it, and we wanted to explain the possibilities that it presents, and we gave it more importance in our slide because of that. Now, for ending, we want to say it's important and actually the fundamental base of science, because sometimes when you start out down a certain path, you never know where you're going to end up in the end. The more research done in the area can really unleash new possibilities, finding new things, different variations of Cas9, or other things that function like the possibilities are endless. To be a little cliche about it, I think it's great for people of all ages to understand CRISPR, at least at a basic level, and a little bit about genome engineering, because so much of the world today revolves around biology and the technology that can benefit from it. Also, we really want to understand ourselves, and CRISPR and gene editing is another way to get at what is the substance underlying us. So we think, without saying is CRISPR ethical or not, we need to do so much more research to make a certain point about it.
Right now, the CRISPR's benefits are more than our illusion, more than our dystopias. So we think right now it is ethical, but we don't know what is going to happen 10 years or 20 years after now. So thank you for watching us and listening to us.
Thank you for your time.
Okay, thank you very much for this great presentation for our students, Başak and Daria, first of all. And that was really enlightened. Okay, thank you very much. Okay, I'm the host right now. Now we will move to the last presenter, Kazakhstan, and I want to try to say hello in your own language, Kazakhstan. Salyametsiz be. Is it true?
It's really great.
Thank you very much, Mr. Kanat. So,
Yes.
I am making you host to continue for this
Excuse me, not me. There is our student named Yeren. Yeren, could you please type something in the chat so that they could see you?
Hello, everybody. My name is Yeren, and my classmates are Bibaris Bakhmohamedov and Sultan Muratbek, and also Kanush Kliulias. So, Bibaris, could you please start speaking?
Hello, dear teachers and participants. Today we are representing Kazakhstan on the topic of CRISPR Cas9. Firstly, we would say, what is it? The CRISPR technology allows scientists to make changes to the DNA in cells that could allow us to cure genetic disease. CRISPR Cas9, short for clusters of regularly interspaced short palindromic repeats, is a family of DNA sequences found in genomes of prokaryotic organisms such as bacteria and archaea. The CRISPR
Part of the CRISPR system is a protein called Cas9 that's able to seek out, cut, and eventually degrade viral DNA in a specific way. A genetic engineering technology, a way for scientists to delete or insert specific bits of DNA into cells with incredible precision that would offer opportunities to do things that really haven't been possible in the past. The CRISPR technology has already been used to change the DNA in the cells of mice and monkeys, other organisms as well. Chinese scientists showed that they could even use the CRISPR technology to change genes in human embryos, and scientists in Philadelphia showed they could use CRISPR to remove the DNA of an integrated HIV virus from infected human cells. Furthermore, there are 6 diseases the CRISPR Cas9 technology could cure.
First one, it is cancer. Using CRISPR, the cells are modified to remove that gene that encodes for a protein called PD-1. Some tumors are able to bind to this protein on the surface of immune cell and instruct them not to attack. The modified cells are then reinfused into the patient with a higher capacity to attack cancer cells. It is how to work CRISPR-Cas9 with cancer.
Second is blood disorders. The first CRISPR trial in Europe and the US, which enrolled its first patient in February this year, aims to treat beta thalassemia and sickle cell disease, 2 blood disorders that affect oxygen transport in the blood. The therapy, developed by CRISPR Therapeutics and Vertex Pharmaceuticals, consists in harvesting bone marrow stem cells from the patient and using CRISPR technology to make them produce fetal hemoglobin, a natural form of oxygen-carrying protein that binds oxygen much better than the adult form.
And cystic fibrosis. First of all, I want to, for those who don't know about cystic fibrosis, I want to tell the definition and explain what it is. Cystic fibrosis is a progressive genetic disease that causes persistent lung infections and limits the ability to breathe over time. And although there are treatments available to deal with the symptoms, the life expectancy for a person with this disease is only around 40 years. CRISPR technology could help us to get to the origin of the problem by editing the mutations that cause cystic fibrosis, which are located in a gene called TFT.
Muscular dystrophy, Duchenne's muscular dystrophy, is caused by mutations in the DMD gene, which encodes for a protein necessary for the contraction of muscles. Children born with this disease suffer progressive muscle degeneration, and there is currently no treatment available beyond palliative care. Research in mice has shown CRISPR technology could be used to fix the multiple genetic mutations behind the disease.
And how CRISPR can save the environment. CRISPR, a tool that can change the lives of billions of people. This specific tool has been earning its name in treating and preventing disease. However, this technique is not limited to the field of the health sciences. Recently, this powerful tool is helping scientists to improve technologies to protect the environment. CRISPR is no more limited to just adding or deleting DNA parts, but could do many more things. For example, if used properly, this genetic tool has editing properties that can increase or decrease the transcription and translation of certain genes. Furthermore, we can also control the rate of expressions of several genes with the help of the foreign transcription factor. These qualities make CRISPR revolutionary tool that can change our outlook towards science. For example, by using CRISPR, we can develop new ways which can help us in tackling the environmental challenges.
Next topic is biodegradable plastics. Apart from fuels, plastics are another factor that pollutes our environment because they take years for proper decomposition. On the other hand, annually thousands of wildlife diseases occur due to plastic ingestions. Therefore, we need organic-based plastics or bioplastics. Certain bacteria produce polydra and microbial polyester. These are bioplastics. Furthermore, we can accelerate the bioplastics production with the help of CRISPR. Recently, CRISPR successfully manipulated a bacterial species, Ralston entropha, for successful bioplastic production. CRISPR can be helpful in accelerating the process of synthesizing plastics. For example, CRISPR successfully hijacked a piece of bacterial machinery for high-throughput bioplastic production. It means the pathways can accelerate using altering genes pathway involved. Furthermore, by using bio-based plastics, they can easily degrade and will not cause harm to the environment.
Alternative to fuels, biofuels. Fuels are one of the most important and commonly used energy sources. On the other hand, it is also a non-renewable source. Therefore, the quantity of fuel around the world is bound to finish one day. As a result, we need to find solution to this situation, and it is biofuel. Biofuel is the cheapest way to generate fuels by utilizing organic matter of biomass. Plants, algae, and bacteria have pathways to develop biofuels. With the help of the CRISPR, we tailor macroalgae for efficient biofuel production. On the other hand, we can also increase the level of efficiency of those cells which already produce biofuels. The precision and efficiency of CRISPR can help us tackle the most complex genetic organism and use them for producing biofuels. Certain bacteria feed upon waste and plant cell walls to produce fuels such as methane. We can use this technology to accelerate the process and help us better shortly.
Reducing global warming. Burning fossil fuels is the main source of carbon dioxide emission. which in turn causes global warming. This global warming will destroy our environment if we did not try to find an alternative. We are dependent on fossil fuels in every aspect of our life. Moreover, carbon dioxide is not the only emissions. Another group of gases, greenhouse gases, is also responsible for global warming. Among them, one of the most potent gas is methane. There are several producers of methane gas, like all ruminant animals like cattle, goats, sheep, and other herbivores and bacteria. The most prominent methane-producing organism is bacteria. Some of them live in wetlands, some live in the guts of the cows and the flooded field rice. We can alter the genetics of cows or the grass they eat so that they can reduce the amount of methane produced. Engineered rice can also curb bacteria to keep as much as methane in the ground as possible. CRISPR can facilitate these efforts, resulting in improving our environment.
Tons of waste is produced annually. Usually, there is no such way of managing this much waste. This increasing amount of waste has a huge negative impact on humans, other living organisms in the environment. Furthermore, in the case of managing this waste, it takes a lot of work and fuel to do so. The fuel is used in turn again harms the environment. CRISPR can solve this issue in several ways. For instance, scientists find out that certain bacteria can reduce waste containing heavy metals. Now, using CRISPR, we can increase the efficiency of this particular bacterium. In a few years, via gene editing up to some extent, will solve this issue.
And also, CRISPR could create plants that yield larger fruit, mosquitoes that can transmit malaria, or even reprogram drug-resistant cancer cells. It is also a powerful tool for studying the genome, allowing scientists to watch what happens when genes are turned off or changed within an organism. It's up to decide the best course forward as CRISPR leaves single-celled organisms behind and heads into labs, farms, hospitals, and organisms around the world.
There's no doubt that gene editing could provide numerous benefits for our society, helping us develop new treatments for a wide variety of diseases and bring an end to some rare genetic disorders. It could also help us create healthier plants that produce better yields and are better equipped to handle the ravages of climate change. Here are some references that we used to make our presentation, and thank you very much for your attention.
Thank you very much, Yeren, Kanar, Bibaras,
Thank you very much. Great presentation, all of you here.
Okay.
Done.
Could I have just one word?
Of course.
As you said before, Salimatsizba is hello in English, and sorry, in Kazakh, and Saubolnas is goodbye. Okay.
Salbozes, okay,
Saubolnas.
Salbozes, but it's not goodbye yet because now we are going to question and answer session together. It's not
great. Okay.
Yeah,
yes.
the party just start. Okay. The party just start. Wait.
Just great. Okay.
Okay. Okay. Now, while you are doing the presentation, all of you guys, all of these countries, while you're on the presentation, during your presentation, I got 4 different questions for you. I took from the listeners first of all, and I just write a question and I will ask one of you guys. I will ask who wants to answer this question, and you can raise your hands online, and I can give you an answer opportunity. Understand? Everyone can see the question in the chat section, but I want to read it all. Read it too. Do you think designer babies can be superiority of a race? Who wants to answer this question? Who wants to answer this question? Okay. You can raise your hands if you click on your names.
Okay, we would like to answer that question.
Of course. Of course.
Okay.
The Turkish team wants to answer the question.
We
Okay.
just discuss are we thinking or not? So we think designer babies can superiority of a race because the beauty imagination in our minds is so basic. Like a woman have to be white skin and tall and blue eyes, and with designer babies we can reach that. So we can do every baby simple and same, like
like products, like they're products from a fabricated.
You couldn't find the correct word, but as an example,
like they're products from a factory.
As an example, like think like Russian girl model. Like when we say Russian girl model, you're thinking a tall girl with white skin and blue eyes. And with designer babies, we think that everyone is going to be the same. Like,
yes, and in society, their imperfections cannot be acceptable, like banned or such things. So a modified generation could claim superiority over another race and not modified race, etc.
So that's it.
Yeah.
I hope so. This technology can find a solution. My weight problem, my height problem too. Okay, so second question in here. Do you think these possibilities may not happen in the foreseeable future, said Giancarlo? So do you think isn't too soon to talk about them? Who wants to answer this question? Turkish team, Malaysia team, Kazakhstan team?
Can I answer, please?
Yes, of course.
Please,
So, yeah.
Kazakhstan team, please answer this question. Please.
I guess the government won't be allowed, will be used by this way, like designer babies, because as the Turkish team said, it will increase of racism. So we know that racism is the most problematic topic at now. So I think CRISPR Cas9 should be allowed by government, like for disease, for increased yields, and etc.
Would you like to add someone? Would you like to add something for this question? Yes or no? Okay. So thank you very much for this answer, Kazakhstan team. It was enlightened. So I'm moving to another question. CRISPR's benefits are too vast to be ignored. How can we overlook all these benefits only for possibilities? Who wants to answer this question? Malaysia team, would you like to add something about this question answer session?
Wait.
Okay,
how can we overlook all those benefits only for
possibilities? Thank you very much,
possibilities?
Dr. Devi, please.
I think when the students presented, we can see there are possibilities, not to say it's too far possibilities. So we shouldn't overlook it. I think we can, we should see the benefits on also the pros and cons of this new scientific discovery. So in that way, I think we shouldn't overlook the benefits. But at the same time, the disadvantages also will be a lot. So it's all up to humans how to make, we have to make the decision. You see, just now, superiority of a race, then the first person said it can cause, racism and all that. I think this one will affect, it for a world peace is not very advisable. Okay.
Understand, totally understand. Thank you very much for this perfect answer. So it's the last question, and it came from the Mrs. Bushra, which is our teacher too. So I'm asking this question for all of you, all teams, because I want to hear your opinion about this one. I'm sending the question on. In the future, do you think society could cast someone out of the community because their body is unable to use CRISPR technology? I really wonder the answer of this question, because it's not only science, it's sociology and anthropology too. Thank you very much for this answers firstly.
If it's okay for everyone, we can answer that question first.
Turkish team, please start. Go ahead.
Okay, we are agree about that. So we think society will do that in the future if this CRISPR Cas9 technology will be used in the human or embryos, will be useful in humans and embryos. Because when you look at a society that everyone has a white skin and blue eyes and tall, when you saw someone who is short or doesn't fit to the beauty mind, or their body is not fitting to use that CRISPR technology, we will actually point finger to them that they're not using CRISPR. They can't use CRISPR because they are different. So
yeah, as we explained, there isn't just one DNA, one genome in everyone that we can use, and it will all be the same result, because us sitting here, we have difference of millions of genes varying from each other. So how can we make something that is that can be used the same way for everyone?
So some genome technology that we can use can be usable in Darya's body, but not in my body. So that will be a discrimination for each other. Like everyone is going to put show finger to that different person. So thank you.
Thank you very much for this answer. Malaysia team, would you like to continue?
Yes, sure. So actually, there is a possibility for the society to cast someone out of the community, because at that moment, when the years have evolved and the mindset of human beings have changed, if the community doesn't have the open-mindedness, they could cast people out because they are different from the others. So if they can appreciate the diversity, then the possibilities for casting out someone out of the community is very less. And another point from my fellow friend, she said that this problem wouldn't affect the community in the future, because not all the genome edited effects can be physically presented. because it might affect our phenotype, but not the it might affect our genotype, but not the phenotype. So it's like a wide ideology, and if the humans are open-minded, they could accept it. Thank you.
Of course, thank you very much for this great answer. And Kazakhstan team, I'm waiting your answer too.
Can I answer please this question? In the future, I think people will choose do you want it or do you don't want it, because it's up to people. It's not like a requirement. It's not like a requirement. If people want to do it, he will do it. If someone don't want to do it, he will not do it. And I think it will be up to people, because maybe someone will not agree with this CRISPR Cas9 and say it is just like a mystery. And I think the people, not all of the people will choose the CRISPR-Cas9. They want their own gene. Thank you.
My pleasure. That was a great answer. So before end, I want a last sentence about this technology and about the INTOC, International Online Class, from all you guys, all these teams. Just one sentence before the end. I want to hear you. Please, Turkish team, you can start. You can start, I think so, right?
Okay.
There is no pressure, no pressure.
Okay. Today, actually, we talk about the chain of possibilities, because all of those things that we talk about will be happen
50, 20 plus years from now in the not foreseeable future.
Yes. And we believe that, as lastly, we believe that now the CRISPR-Cas9 technology is ethical, but we don't know for sure if it's going to be ethical for 5 years from now.
Yeah, before discussing the ethical reasons, I think we need to make a lot more research and understand what CRISPR actually stands for.
Yeah, if we ban this CRISPR-Cas9 technology now, or we just stop the research about CRISPR-Cas9, we will have question marks in our minds.
Yeah.
So thank you for listening to us, to everyone who is participating in this discussion, and thank you for having us in your homes.
That's a weird way to put it, but yeah.
Yes. Thank you very much.
Okay, thank you very much to Turkish team to attending this presentation. Again, thank you very much, guys. So Malaysia team, last sentence before go.
Okay, CRISPR-Cas9 is a unique technology. It is well known for the flexibility and efficiency of its editing system. So it has opened up the possibilities in the gene manipulation, gene knockdown, and gene knock-in. So mostly it's not more preferred to the system because of its cost, and it's most probably cost around like $1,000 to carry out this system, and its success rate is around 1% based on the gene insertion. And that's all from our team. Thank you.
My pleasure. Thank you very much, Mr. Autas. So Kazakhstan team, last sentence.
Yes, thank you very much, if you allow me. So as concerning the CRISPR-Cas9 topic, I would like to say that, as my students stated, that there are lots of benefits from this technology. And well, you might think about it as the nuclear power, right? So when got in a kind of, well, you say bad hands, right? Bad people, it can be used as a as the weapon, right? But if it is used correctly, it can serve humanity in some, positive way to, provide people with energy. Well, if we talk about CRISPR-Cas technologies, it can provide people with like, new possibilities, let's say so, right? Concerning the INTOC classes, I say, guys, it's just getting better and better every day. And I say that that's just a great opportunity for all of us to experience that unique opportunity. Just don't give up. And well, let's just make it better and better. Thank you very much.
Thank you very much. So thank you very much for all teams and all you one by one to join us today. Don't forget, our next meeting will be 14 November, and we will be together with the real valuable professor together. So thank you very much to attending and joining us today with us.
Excuse me.
Thank you very much for touch.
Excuse me.
Yeah.
Excuse me, host. 14th November is, can you hear me?
14th
Hello?
November.
14th November, is it?
Yeah.
14th November is our Deepavali. It's our main festival.
Yes. Okay,
So
maybe you and our supervisors will talk about the date later.
okay.
But I'm checking my dates right now, schedule right now. It's 14, so maybe we will talk about later.
Okay, because we want to participate.
Information about the next meeting. Thank you very much for joining us. Thank you very much from Turkey, all of you guys. Thank you very much. Thank you very much. See you later. See you later.
Good job. Thank you.